you all there for this very important topic that is reproducibility of our research. Well, the first time José told me about that, I thought that we had something that is very important, especially for our young folks, because well, the basis of what we do is should be based, if we want it to work, on an excellent quality of our research. And we know since a long time that the question of reproducibility of research is something that is tough and always a matter of challenge. Well, I'm the director of the Oncosphère Nouvelle-Aquitaine. I know whether you know what is the Oncosphère Nouvelle-Aquitaine. That's a structure that includes all the researchers working on cancer around Nouvelle-Aquitaine. That includes the main towns, Bordeaux, certainly, but also Limoges, Poitiers, La Rochelle. It may be Pau, but Pau is not inside currently. We have to federate all our initiatives in different domains and disciplines. That includes biological research, certainly, translational and clinical research, and also all the fundamental sciences, physics, mathematics, bioinformatics, and so on, and also epidemiology, public health, sociology, communication, and so on. So that's very large, and the objective is to improve our management of cancer and the way patients will live, support, manage, and be treated for cancer. So I think now that's a moment we have to spend most of the time to discuss about reproducibility, and I would like first José Eduardo Gomez to be the first one to introduce the meeting, because he is the one with others that have built this meeting. Thank you, José.
So thank you, Pierre, for your kind words. So I'm gonna start by thanking Pierre and Oncosphère for the support you gave for this organization on behalf of the committee, the organizing committee. I'd also, of course, like to thank Candice and the people at Consulté, Virginie and Elisa for the support to the logistics. They've been a great help. And I'd like to say a few words about this workshop and where it comes from and what we'd like it to be. So I'm very happy to see so many students here today. Like Pierre said, I think it's mostly for the new generations that this is going to be useful, I guess. So this the issue of reproducibility in science, and particularly in cancer biology, it's something that we've been worrying about in our private conversations for quite a while now. It's probably at least 10, 15 years or so that we've been informally discussing constantly on our conversations. It's something we worried about and we kept in the back of our minds for a long time and actually the history of this workshop, it's directly linked to the papers that came out on eLife 2021, in which Tim Errington is the main author. And then decided, no, we have to do something about it. I don't know what we need to do. And I was over coffee in the morning with that time actually. And he told me, well, you know, Maël, who's on the organizing committee, is also worried and thinking about those kinds of problems. And so I reached out to him, started talking, and then immediately we thought about Masha as well to join us on the committee. And then when the three of us discussed, we need a fourth corner on this thing, and we wanted to give also a regional dimension, but have a different perspective. And Mathieu was actually the first name that was mentioned, and he also accepted to be part of the committee. So the first thing that we should notice is that we come from very different fields related to cancer. So I work on cell biology, cell division with a genetics approach, basic cell biology questions, more on that later a little bit. And Maël is a philosopher working on cancer, among other things, from philosophy of medicine perspective. Masha is a bioinformatician working with sequences and algorithms and big data. And Mathieu actually works with ecology and evolution and these funny transmissible cancers that are swimming in the sea. We all have actually quite different perspectives on what this issue of reproducibility is and means. And so we try to put that into the program to have an interdisciplinary approach and discussion of that, and to try to integrate these different perspectives. So interdisciplinarity was there from the beginning. And then we started, like each on our field, thinking of people that we would invite. We were quite lucky because the first choice people accepted to participate. And so we have a very nice lineup. I don't know if you have the program there. We have 4 excellent speakers. A very nice lineup. So Timothy Errington is the main author of the papers of eLife that I just mentioned, that did like led the biology reproducibility initiative at the Center for Open Science. Then we have Marcus Munaf, who is actually going to be online for logistical reasons. We couldn't find a way for him to travel because he has a busy schedule and the traveling was complicated. But he's actually has been in the forefront of the reproducibility crisis discussions in psychology. And he's been like actually, by the way, psychology, they are like 10 years ahead of cancer on these aspects. And he's actually more recently collaborating with the UK Cancer Reproducibility Network. So he has something to talk about on that, but he's the chair of the UK Reproducibility Network. And then we have Frédéric Delamotte, and actually it's the reflex of our interdisciplinarity. He's not actually working on cancer per se, which is actually, it's difficult to find people that are working on reproducibility and cancer. It's like not enough people working on that. And he's going to be talking, it's a bioinformatician, he's going to be talking about open data and shared data, which is very important. And then we have Don Thierry, biostatistician, who's going to be talking about statistics, how we working in biology, biologists can interact with statisticians and can make proper use of statistics. We need some of that, a lot of that. And then I'm going to try to, I'm not an expert on reproducibility, nor I pretend to be, but I'm a biologist working mostly at the bench, working with genetics, and I'm going to try to present the point of view of somebody doing experiments and somebody that actually should be worried about reproducibility, things we can do about it. So we managed to get this lineup. We were probably a bit ambitious at some point in our discussions. We discussed a lot about other people that we should invite and what the format of the workshop should be. Actually turned out quite well. There was actually one change recently in the program you might have noticed, because we had one more person in the domain of philosophy that finally couldn't make it here today. And that kind of solves a problem we had with the organization, because we wondered a lot, should we have like a final discussion, an open discussion in the end? Round tables sometimes work, sometimes they don't. So in the end, we had one more slot at the end of the day. That is going to be a slot for discussion. So we're going to have 1 hour for open mic for you to ask questions. So it's going to be probably a great opportunity if you have some questions that you would like to ask several of the people talking today and have them dialogue and interact with one another. That would probably be the time. We're going to have, so the idea is that we have fruitful and useful discussion in the end. So this issue of reproducibility, as much as it's kind of something to be maybe worried about, I think it's actually an interesting, and I think I can speak for all of the organizing committee. It's an interesting scientific question in itself. So we hope that we can have not just stress about all the research that is not reproducible, but actually have a questioning of the research we are doing. This is the opportunity to ask ourselves, what are we doing? What is science? What can we do to make it better? How does science work? This kind of very fundamental questions about our job as scientists that hopefully in the end will also provide some guidelines, some way forward. But the goal is basically to have as much discussion as possible. So we made this program with plenty of time for discussion. After the talks, during the coffee breaks, at the end of the day, lots of opportunity to discuss. We made it so that there's time to ask questions and give your opinions and give your points of view. And then hopefully by the end of the day, we solve the problem of cancer biology reproducibility. So that being said, I think we are on time to move on to the first speaker. So like I just said before, but, Timothy Errington is going to be the first person speaking. He's the lead author on the papers of the cancer reproducibility project that came out on eLife at the Center for Open Science. And he's going to tell us about the history of that project, the results, and the recommendations going forward. So without further ado, I would like to thank specifically all the participants, the invited speakers that made the trip to Bordeaux and give your time to be here, being the first one to speak. Thank you, Tim, thank you, Don, thank you, Frederic, for being here today. And we can now move on to the first speaker, Tim Errington. Thank you.
Well, thank you for the invitation, first of all. I think this is a great idea. I'm really hopeful that I can set the stage a little bit in terms of what the rest of the speakers will elaborate on. So let me give a little bit of background about the Center for Open Science. I don't really have those slides, but I figured there should be a little context. So the Center for Open Science is a nonprofit based in the United States. It's in Charlottesville, Virginia, so where the University of Virginia is. We're just actually reaching our 10-year anniversary this May. We're really excited. We've been around that long. One of the things I want to echo here is that the center, the director, Brian Nosek, he's a psychologist by training, a social psychologist. So when we think about this being multidisciplinary, I now get to live and breathe that, and I love that. That really looking at it across disciplines is really important. And what psychology has done doesn't mean it perfectly translates over to what we need to do in the variety of fields of cancer biology, but there are lessons learned, right? And that's true for many other fields. So I really enjoy that as a part of my work. And that's a big part of what made COS, I think, unique and special. This is a final note before I get into my talk. The center, I didn't put it up here, we're a nonprofit, and just wanted to disclose, like, we're largely funded by government foundations, so no different than other researchers, right? We're soliciting funding from national funding agencies like the NIH in the US, but also a lot of private philanthropic groups, as well as private donors as well. Okay, so today I want to give you the journey of what I've been doing at the Center for Open Science, or what I was doing for the last 8 years when I started at the center in 2013. Basically, this project that we wrapped up took about 8 years to wrap it up at the end of 2021. And when I was a grad student, I think, speaking hopefully to some grad students in the back, that's when it really hit me. I was having issues myself with trying to reproduce experiments in the lab I was in. So give me an example of that. When I started in the lab that I was doing my PhD in, the very first thing I did was to see if I could replicate findings that were just published by another researcher, another grad student in the lab. I couldn't do it. I had the paper that didn't give enough details. I'll get into that in this project. I had the person, they didn't really remember everything. I had all the notes, right? And it showed me the inefficiency. And I eventually figured it out. It took me about 6 months to figure out exactly what they did to get back to the same starting point that I wanted to start at. I thought I'd hit from day one. And so that really resonated with me. I was like, oh my gosh, this is so inefficient, right? Why am I spending 6 months just trying to do the same thing when I have access to all these resources? And how is anybody going to possibly do this if all they have is the paper, right? I had access to all this other information. And that's not suggesting anybody was doing anything wrong, by the way. That's just the state of affairs. At the same time, there were papers that were coming out, white papers by pharmaceutical companies, Bayer and Amgen are the two that I have up here. And I still remember when those came out, 2011, 2012. They caused a lot of noise because it was, to me, did two things. One is it raised the alarm. Hey, we're having problems. We can't replicate research that's coming out of other labs. But it also felt a little finger pointing. Somebody's doing something wrong, right? A little bit pointed towards academia, which I don't think was their intent now that I've talked to all the authors. And the thing that really stuck out to me is when you read those papers, because they are white papers, the evidence naturally is limiting. This is it. This is all they tell us about what they can't find, right? About 20 to 25% can't replicate, right? About 11% can't replicate. So they don't tell us the papers because they can't, right? They're not allowed to from just like disclosure agreements. They don't tell us what they mean by it in the definition. They don't show us what they did, and they don't show us their own results and how they actually assessed it. There's no knock on them, right? They had to do it this way. But this was essentially the spark for the project, right? Which is there's something going on, we're having a hard time. Somebody's telling us there's a problem, but we don't know what they're talking about, right? And so this was essentially the spark for the project that I ended up leading at the Center for Open Science. It was also modeled off of the psychology one. And the reason why, and I'm gonna pull up a little forward here to an informal survey done by Monia Baker at Science, or Nature, I mean, which is that this was a concern by many researchers, right? It wasn't just an isolated incident. So this is, you probably have seen this, right? It's a reproducibility crisis, and it says, you know, informally surveyed about 1,500 researchers that many say there is. Just as a note on this, I don't actually love using the word reproducibility crisis. I much prefer thinking of it as an opportunity, right? This is part of the process. So I'll come back to that, but I think we have an opportunity in front of our hands. But the thing to highlight is it's not disciplines, right? It's not just one, it's not just medicine, it's not just psychology, it's not just biology, and it's not just somebody else's work, it's your own, right? This is to me the really thing to think hard about, right? Which is what does it mean to not have irreproducible results? And what do we do about that? Okay, in 2014, those were after those papers were published, right? After we decided to start this, we got funding, not from a large funding agency, but a brand new philanthropic group called the Lawrence John Arnold Foundation. They're called the Arnold Venture Group now. The reason, by the way, they funded us is they were not interested in cancer biology. They were interested in how do they, as a philanthropic group, know where to invest their dollars, right? There's lots of great causes out there, but when they look at the research behind it, across disciplines, they see concern. So they were actually more interested in research integrity than they thought at the start because they wanted to know where to invest their dollars. So they invested in this project because they said this is exactly the type of thing that we need to invest in, which is, yes, we want to invest in cancer therapies, cancer patient treatments, but we want to also make sure that we're putting those dollars to the right use because there's lots of opportunity out there. We also did this in partner with Science Exchange, which is a group that's based out of Palo Alto, California. That's a bit of a marketplace. I don't know if you've heard about them. You should check them out. They're a really interesting group. Essentially, pharmaceutical companies use them as well. When you're looking for expertise of, say, trying to do like mass spectrometry or cell biology experiments or animal studies, you can find experts that are largely CROs, contract research organizations, or academic core labs. So that essentially meant that they were our partners to find the researchers, right? We outsourced it. And the last partner that you've heard is eLife. We found a journal that was willing to publish our results, and I'll show you the model we used, but also hold us accountable as researchers. We kind of needed to make sure that we use the peer review system, I think, in this case, to its advantage, which is helping us be and do the best job that we can. All right. When we started this project, we weren't interested in trying to figure out like, oh, there's a concern with X paper. We were more interested in systematic sampling. We looked at papers from 2010 to 2012. We did this in 2013. And we basically just looked at which ones were making the most attention, which one were people downloading the most, which ones were they reading the most, using things like Mendeley Web of Science. And we look on different search terms, and all of this information is available online if you want it. What we didn't do on purpose is exclude a lot of the large omics research. The reason why is that's just a different beast. There have been different projects, and I think there's a lot more work that can be done there. So we avoided any genomics, proteomics, or high throughput assays, or anything that required like a novel technique or a novel research reagent, right? Something that we know we couldn't get our hands on. So what we're really looking for is your cell biology, your biochemistry, right? Your molecular biology, your kind of bread and butter. And when we first started this project, I thought I'd be standing here telling you what we did in psychology, because there's a similar project, which is what did we find? What are the outcomes? But today I'm actually going to spend more time talking about the process, because the process actually, to me, I think exposed a lot more than what the outcomes are. Before I do that, one key feature that we put into this project that we did with Eli is we thought about how to do the replications. Typically when people do this, if they do them at all, is they do them by themselves, right? They read some paper or they find, like what I just did. And you go off on your own and you try to piece it together. And what happens is as you get your results, there's always hindsight bias, right? Somebody says, oh, well, it didn't replicate because you didn't do X, Y, Z. And you're like, oh, okay, I didn't know I should have done X, Y, Z. You didn't tell me to do X, Y, Z. So what we did is we did something that we think of as pre-commitment, which is figure out and get all of that information done up front. And I'll show you what that means a little bit. We did that in the peer review process, which is before we begin any of our experiments, we really are kind of registering, why do we think this wouldn't work? We should have no a priori reason to think this is not going to succeed. That was our intent. I wanted, and I still believe when we started every one of these projects, there was no reason it wasn't going to work. So when it doesn't, that shows us something. And so that was this key step. And what does that mean? I'll show you in a bit here. This is slides I'm borrowing that kind of does a nice overview of kind of our overall process. A pointer here. So at one, we started with these papers, identified a little over 50 papers. Then we identified what are the key experiments we're going to replicate. These are huge papers, by the way. These came up to be Nature, Cell, Science papers for the most part. So dozens and dozens of experiments. We weren't going to do all of them. We were hunting for what are the quote unquote key ones, again, excluding those genomics experiments that were in the paper. What we meant were, what was in the abstract? What was a key claim that was being made in the abstract? And can we find experiments that support that, right? That include our inclusion criteria. So that's what we started with. Then we identified those experiments and we started with the paper, just like I told you I did with my grad work. Go to the paper, go to the methods. I'll get into that in more detail because if you guys still try this today, it's interesting to see what happens. Then we go to the authors, corresponding authors, and try to see if they can help us, because usually there would always be questions, and that's okay, but sometimes there'd be a lot of questions and sometimes there'd be a few questions. And so we'd see if we can get some information about understanding the experiments, getting access to the data, getting access to reagents. There's some reagents we couldn't get. And after we did that, after we attempted to go through that process, we also identified labs from Science Exchange, and then we wrote up what we're going to do. So we spent all this time not doing our experiments, right? In terms of at the bench, all this time thinking about our experiments, trying our best to design the best experiments with hopefully the original authors that were the ones that did it the first time. And we sent that to eLife. And then the peer reviewers, the original authors again, biostatisticians and other experts in the field, they weigh in on this design. Not what we found, but what we were designing to do. And only after that finished peer review was accepted, after revisions, then we began our experiments. And then after that, we would publish all of these papers one by one until eventually we kind of ran out of time and funds, which was great. And then we stopped the project. So what I'm going to talk about now is in summary, not any individual paper, but in aggregate, what did we learn? And these are the papers we published. I published them in 2 papers. And the reason why is because I think there's 2 stories going on. One that I'll talk about first, which is the process. And then the one I'll talk about the second briefly, which is what did we find for the ones we could complete. So let's think about the challenges, the process of doing this. So again, if we think of this in aggregate, I'm going to do this at experiments. There's 193 experiments that we were attempting to replicate across 53 papers. Experiments to me is the right unit. Each step along the way, we ran into some challenges. First is data. So why do we need data? We need data because I want to know what the effect size is. I want to know how many replicates, how many animals we're going to do in the replication to be able to actually see if we can find, an effect of interest. We need the original data for that. Very few, 2% had open data, right? 16% shared after request. So basically what that means is I have no data. I have not, it's only what's in the paper. And what's in the paper tends to not be the actual raw data. It tends to be representations of it, a bar graph with error bars. It doesn't tell me anything about the raw data behind it. I have to make assumptions. I can get past that, and we did, right? And I'm sure we'll talk about that a bit later, which is the challenges we have when we do this. But now I'm stuck not actually understanding what's the original data, what was actually found, what are each of the data points, and how do I make sense of that myself as an independent researcher? But we moved on, we made assumptions, hopefully correct, probably incorrect. Similarly, when there were statistical tests, not always done, representative images are pretty common. But when there were 21%, we had no idea what they were, just an asterisk, p less than 0.05. Sometimes I wouldn't even know what the comparisons were. You'd read the paper and be like, what are the 2 groups they're comparing? Like wasn't quite obvious because they'd have a lot of different groups in there. So this is a challenge. And again, this is just interpretation. I can't understand entirely what's being presented in the paper. I don't know what tests they did. I don't know what the comparison groups are. And even when we asked for code in these cases, very few had it. And I'm talking like Excel sheets, Prism files, anything. Very few had it. Only 13% were able to share the code. So again, we can make assumptions. We can read into the paper. This is why actually our process helped asking the authors, asking the peer reviewers, tell us what we should do because it's not in the paper. We want to make sure that we're doing a good faith effort. So what are the right comparisons to look at? What are the right sample sizes to look at? Again, I can work through this when we did, right? We hopefully get information. A lot of times we didn't, as I just showed you, or we'd make assumptions. And then we'd have those tested by other people independent from us. Where it got really tricky was reagents. So here I'm talking key reagents, cell lines, animal models, plasmids, antibodies, right? That's your workhorse course right there. And I'm talking about reagents that are not commercially available. I can't go to Sigma to buy, or that are not ideally in a repository, right? Like AdGene or ATCC. Here we have to ask them. So a lot of the times we had to have at least one unique reagent of that type that I just told you. And we asked for 70% of those required for that, but only 69% were willing to share. So that's a barrier. Actually, I was just talking about this earlier, right? Which is, isn't it great when somebody asks you to share? And they do an amazing job of really giving you the reagents that you need. Do you know how frustrating it is when they don't share, right? I've actually experienced that again as a grad student. And here we are doing this in a project that we knew would actually get more attention. So this is probably the rosiest picture we have. And this is something that we know we can get past. There are great groups out there. One can share with each other, but there are also repositories to reduce the administrative burden of it. So this is, I think, a low-hanging opportunity. What did it mean for our project if we couldn't get the reagents? Well, I had to find a different reagent, or I had to remake it. That's the one that really did this, remaking the same thing. I'll give you an example of how extreme that was because we couldn't do it. There was one paper we were looking at that had a triple transgenic knockout mouse. That was the model. They published it in Nature. We asked for an animal. We're like, can we have that animal? You just published it. They're like, no, you remake it. We're still using it. We're trying to publish more papers on it. We're like, there's no way we can sit there and invest the years and however many dollars of research to remake a reagent you just made for us to test it one more time to validate that. That's the challenge we had. So we dropped that paper. So this is what happens is we start to drop things as we went through the project because we weren't sure how to navigate it. And one time it got so bad that we did eventually interview with the journal and the authors did eventually share us. That's not really what I wanted to do. That's a great way not to make a friend, which is to have somebody intervene. So we should think hard about our culture, right? Which is when somebody asks, what do I do to reduce the burden on myself and them? That's why I think open repositories are a great thing. But again, I'll come back to that at the very end when we're taking recommendations. And then protocol clarifications, like I said, we had to ask information. So one, we always had to ask questions. All right, that's fine. There's always details, right? The devil's in the details. But to me, what stuck out more was the degree. Sometimes about half, it wasn't too bad, right? Sometimes it's like I flicked the tube versus I spun the tube. And if that's the huge difference, then you better tell me. But that's not something we'd be asking for. Sometimes we'd be asking for incredibly important reagent details, like how long did you treat the animals? What did you assess the animals? What was the strain of the animal? Huge details that were lacking in the paper. So it varied wildly in terms of what we had to ask. And again, if we didn't get information, we'd have to just make assumptions and move on. And we'd ask, and this is, I think, the more interesting one, which is it splits, very bifurcated. Half the time we had incredibly helpful authors. There were people that just did an amazing job. They'd hunt down and find, and they were all willing to show us. They'd give us, information that they couldn't put in the paper for whatever reason. And they pull out those details, hand us those reagents, and give us all the information that we needed to be able to proceed. And then sometimes we'd have people either not respond to us or tell us, you know what, I'd help you, but you're doing a replication, so no, I'm not going to help you. So I think this is the thing to think about. I'm not trying to sit here and shame. I'm talking about this is the culture. This is actually back to your point, which is we should be talking about this. This should not be something that should be seen as a threat, but I get it. It's still a threatening topic sometimes. Okay, let me say what I just told you, and I'll show you it a different way. Here's an example paper. So that first image, see if this works. No, that first image here on the left, that's just your main figure, right? Bar graphs, representative image, asterisks. It's pretty common. We still do this. That's your methods in the supplement. Very sparse. And it usually does, and I think we still do this, and there's nothing per se wrong with it until it does lead you down a trap, which is it doesn't actually tell you everything. It points to another paper. And actually that example I had from my grad work did the same thing. So the method is in a paper that is referenced. You go to that paper and it references another one, and another one, and another one. Then you're like 20 years in the past and you can't even find the method that's going on anymore. So that's the challenge we'd have, right? And we do that because we're writing our methods after the fact, right? We're more focused on our outcomes. We've lost focus on the process of what we found. So we'd compile it. On the right there is just a representative images of, we'd send them Word docs. We'd highlight the questions we had and we'd always say, tell us anything that we have wrong and anything else that you think is important to make sure that we're doing a good faith replication. We want this to succeed. And like I said, we'd have varying questions, right? We wouldn't know what antibodies were used, right? The sex, like I said, the sex of the animals. We wouldn't know like how even certain outcomes were measured. It's just a positive area. So lots of way to define positive area in a representative image from microscopy. And we'd always be asking them for more information. What was the protocol you used, right? Standard protocol is great, but if the devil's in the detail, it's not great. So tell us what you believe are the necessary conditions to get the same result. Like I said, sharing data and sharing other things was the key part. This to me is just a summary, and again, it's not verbatim, I'm not pointing to anybody, but this actually highlights, I think, what the challenge is, which is it's not per se always people wanting to not share with us. They were there. What's more common is we weren't set up for it, right? Oh, right, the paper, when I wrote it and I sent it to the journal, they made me cut back on my text. I only had so many words. You know how many times I've heard that? So many times. I had people telling us, oh, you know what? Here, let me give you an earlier draft of that manuscript because the journal made me drop figures that actually are relevant to your question. So they share me earlier drafts. This is actually why preprints, I think, are amazing. I'm not talking about that, but this is an example of how it helps. Data, publish the paper, data goes away. I essentially heard the equivalent of the dog ate my homework, right? There was one example of somebody who did their work as a postdoc. They moved, started their own lab somewhere else. We eventually got put in touch with them. They were like, oh yeah, I was like, we do have the data. Go back to my original lab. They have it, right? Because they're the ones that are supposed to keep it. We're only what, 1-2 years out from publication. And they said, oh no, when they left, we just erased it. It was on the hard drive. We just erased all the data. So it's gone. So even if I wanted it, it's all gone. And it's just because we're not set up, we're not thinking about preservation, not thinking about, well, what else could someone do with that data, right? I published my paper, that's sufficient, but really it's not. That's the start of the conversation. And it's also done because we collaborate. People move. So yourself, right? Like we will leave. You will go when you finish your work and you'll go off to another lab. And so your job is to leave a trail for someone else to be able to pick up on, someone that you don't know. That might be your future self, or it might be the next grad student or postdoc that's coming in behind you. And basically what happens is we just don't have things readily accessible. So that's what I heard is it's not, I don't want to all the time. It's that I can't, even if I wanted to, I can't help you. All right. We did do some experiments. We did less, hopefully you understand a little bit why. And we still ran into some issues. One, going back to that reagents is not everybody actually shared with us, even when they said they would share with us. So I told you one where we had to intervene with the journal. There were other times when we didn't. So we'd have to make these decisions. So what are we going to do? Do we bug them? Do we get the journal involved? Do we really want to go that path? Or do we remake them? Or do we stop? So there's just a bit of this is a bit of a culture. And it also, again, shows you that there's a barrier we can get around, which is using reagents and depositing them in open resources. And well, there's two things here. One, we had to modify our experiments. Surprise, surprise, right? Anyone who's done an experiment at the bench knows that it rarely goes according to the way you want it. What's surprising to me is actually it did. Our process worked. What I'm talking about here is our controls work. That's what I'm referring to, not the outcome. I'm saying our controls, our positive and negative controls worked. Our model system behaved. What I find surprising is that we did that. We went through all that work and we actually had a good chunk of them proceed. Now the results didn't come out the same. That's a separate question. But our experiment worked, right? Our control, our model system behaved. Sometimes it didn't. This is one spot. If I had to go back and do this again, I think harder about this, which is we should have anticipated more of this going on, which is how do we actually optimize? We don't always know what's going on in our own lab. So when we try to transfer it to somebody else in an independent lab, there's going to be instances of information that are lost that we didn't realize. And that maybe we should be focusing more on that instead of outcome. That's to me, and I'll come back to that as like, that's the big surprise to me, which is we should probably spend more time thinking hard about what does it actually mean to get the result? What are the actual conditions we need? But we would have to modify sometimes very minorly, which is just optimization of, say, transfection techniques or infection techniques. And sometimes it would have to be incredibly dramatic, which is the whole model system isn't behaving. An example of that would be one where we had a lymphatic lymphoma model in a mouse. Basically had the authors share us everything. They told us what to do. We set it up. The model system behaved completely different. The timeline was way, way, way off. We talked with the authors. They basically were like, well, shoot, okay, why don't you just try modifying a lot of this? We ran some pilots, eventually found a window that worked, that wasn't perfect, because eventually the animals would progress so far in their disease, we couldn't actually detect what we wanted, but we were able to run the experiment for the most part. That's been a lot of resources. We had to go back and forth with the authors who were great. We had to modify a lot of key aspects in terms of our system. And in the end, we were able to get a result, but it didn't work perfect. That's just the control, that's the model. And I think there's a lot to be said about us thinking hard about not what is the result I found, but what's the model system I'm using? And comparing that to other groups. When you compare some of these animal models to other groups, you realize they're wildly different from each other. And when we think about that, that should be something we should really be thinking hard about, because we want these things to translate to the human domain, and none of us humans are the same, and we see wild variability there. So let's start embracing variability instead of trying to be afraid of it. And we were able to implement all of them, and I think the reason why is because of what I was just hinting at, which is it was hard, right? We had a finite amount of resources. If we had infinite resources, we could have kept going. But we had a finite amount of resources, thankfully. We had somebody that funded us. So eventually we had to stop this, not implement them, or we just couldn't figure it out with the authors. We were just like, yeah, we don't know. We're trying everything you're saying. We still can't figure it out. So at some point we just kind of put our hands up and said, we don't know what's going on, but we're going to stop expending our resources trying to figure it out. So it's kind of left in limbo. All right. I didn't show you any results yet. That was just our process, and that's the big part. And the issues we had were really about communication, as far as I'm concerned, and transparency, right? Which is not having enough accessibility and transparency of information. That was actually the biggest barrier that I encountered. Not having access to data, to reagents, to the protocols. That was it. And there is an element of being dependent upon communication with the authors, which is a downside if they're not helpful, and also recognizing that that's not what science is about too. At least I don't think it is. It's not about me, it's about what I found. And so my job should be, how do I transfer that information as best as possible to somebody else? And naturally, we just ran into these resource questions. We don't, as a grad student and beyond, I never really thought about resources the way I did in this project, which is here I had a very finite budget for a very specific project. And we would document everything and watch how far we could get with everything. Versus I think when we do it in academic research, there's a lot of it of, I have resources and I'm trying to pursue work over time. All right, we did find stuff. And the reason everything I told you I think is important is because when we did actually get outcomes, they didn't always match up. And so I think that's actually why all of these challenges are important. What do I mean by that? Well, one is, what does it mean to replicate? So this is the slide I showed you in the beginning with Bear and Amgen. They throw some numbers up, we throw some numbers up. Here's 5 different ways from a statistical standpoint to look at replicability. Simple things like, is it in the same direction in p less than 0.05? I don't think that's the best way, by the way, but if you do that, it looks pretty rosy at 47%. If it's the original effect sizes and the confidence interval of the replication, I'm a bigger fan of effect sizes, then it looks not so rosy at 25%. And there's other ways we can do this. There's other methodologies that we can use, combining the experiments in a meta-analysis. What you get is a variety of methods that have varying degrees of how conservative or liberal they are, all of which I think are wrong. And maybe some of them all are right, depends on the situation that you get. What we decided to do is to say, well, we don't really care about any of those individually. Those are just methods that people have used that you can get at it from a quantitative standpoint. What if we just look at how often they succeed on all 5 or 1 or 2 or 3 or 4 of those, regardless of what type they are, right? So I showed you 5 different ones. If they succeed in all 5, how often does that occur versus less? Just as a note, when we were doing these key experiments, we wouldn't just pick positive results. Don't get me wrong, the literature is full of positive results, right? The drug shows some efficacy, but we would also include null results if they were important. For example, the drug shows huge, increase in efficacy based on, say, tumor-free survival. But it also shows that there is no damage occurring in the heart. That's an important thing, right? So we would include that null results as well, so doing sensitivity analysis. And what you see is when you look at this, irregardless of those 5 criteria I showed you, that if you wanted me now to sum it up into a number, I'd say a little under half succeeded more often than they failed. Okay, this is, by the way, how we're operating now. So one can look at this and say, oh my gosh, there's a crisis, right? It's a flip of a coin. I'd say, imagine, look how much we're doing, knowing that this is what's happening and all that inefficiency I just showed you. Now imagine if we could start working on that and improving that efficiency. Imagine how much more we could do, right? So thinking about optimizing our research enterprise, essentially is what I'm getting at. So what else did we find? Well, to me, effect size is kind of the biggest thing, right? Effect size being right, like that mean difference between, say, control and your treatment. What you see here are 2 graphs. One's zoomed in on the right. And to me, this is kind of the takeaway. You have effect size of the original on the bottom right. The y-axis there is the replication. So in an ideal world, if I did a perfect replication, everything would run up on that diagonal line. right? A small original effect would have a small replication effect. A large original effect would have a large replication effect. And what you see is things are hanging out south of the line, south of that diagonal, which basically is telling us that on average, 85%, the replicants are 85% smaller than the original. That's staggering. So let me give you that as an example. Animal model, 20-day survival, right? Increase with the drug. 3 in a replication. 20-day 3, right? That's what we're talking about, right? So when we think about our findings in the preclinical space translating, I would think, wow, 20-day survival in animal, that's amazing. 3-day, I don't know, is that as amazing as I thought it was? So like that's why I think we need to think hard about this is we want robust effect sizes that we will persist beyond the preclinical experiment models that we're doing. If we look at it another way, this is just looking at the effects. Again, all the data on the left, just those positive findings, just those null findings, right? The original there, you see, by definition, everything is above. This is just a distribution plot that we're seeing. And our replications, we see that the median comes way down, right? We still see some high effects, but by and large, the average sits closer to 0, which basically says there's no effect. And if you like p-values, I don't, you see the same thing, right? Originals are largely all below less than 0.05, that magical cutoff. Someone will probably get into that in more detail. When you look at replications though, it's a lot different, right? All right, one more thing before I kind of give you some summary, which I hope I'm okay on time, which is another key part of what we were doing was to think about what are the factors that are involved, right? So yes, there are challenges I just showed you. Yes, not everything's mapping up perfectly on a diagonal line. I don't know if it should, by the way. But are there any things that are important? One that stuck out to us, there's a handful we explored, but one that was really important for us was animal experiments, right? I think animal experiments were considered to be a little bit more challenging because there's more variables at play than, say, non-animal experiments, the tissue culture. All right, so we tested that. And what you see here is animal on the left and non-animal experiments on the right. That same graph I showed you. And what you see is when you look at some of these metrics, you're nervous. You're like, wow, only 12% of the animal experiments replicated when we think about significant in the same direction compared to 54. That's a dramatic difference, right? 12%. But what we see is that actually the effect size differences are the same. So to put this another way, we actually don't think this is due to animal experiments. We think it's due that animal experiments tend to have smaller effect sizes and that the smaller effect sizes themselves are harder to replicate. If I'm saying that with the limited information we have, this is something that if one wanted to really follow up, I think this would be a follow-up area to do more research and reproducibility. All right, one more thing we didn't look at, but I'm going to highlight here because Jocelyn Kaiser at Science did this, and she put this graph together with her colleagues at Science. But eLife was essentially doing this in the background, which is subjective assessment, expert opinion. That's one way to think about replicability. What do you think, expert scientist? Well, we were having that essentially happen at the paper level by eLife. eLife editors and reviewers were essentially reviewing all of our work, our pre-proposals and our final results. And the editors would say what they thought. I don't agree with it, but this gives you one viewpoint, right, which is important. And they've kind of been it off of 4 categories, right, whether they thought it was positive, the same, null, right, didn't work, did not replicate, as negative, mixed. Some experiments did, some experiments didn't, or they felt that they couldn't interpret it based on whatever we found, whatever challenges we ran into. So this is just another way to view it. And depending again how you decide to slice this, you will get a glass that looks rosy or a glass that looks not so rosy. So I think I'm being unsatisfying in the sense that people want to say, Tim, how many replicated? And the answer is, what do you think? And I think that's unfortunately what we're always stuck with. And the more important thing would be, what does it mean to replicate for you for that given experiment? All right, so some conclusions and then some kind of takeaways for me. One, right, effects were much weaker than original. So that's why those challenges are important. And depending on how you look at success, right, like I said, glass is rosy, glass is not so rosy. And you know, the animal experiments, as much as they're important, I think there's room to investigate further, but we actually think it's more about small effect sizes, small differences. Maybe the bottom line is I think there's just room for improvement, right? That decrease in effect size is to me the most staggering, 85% smaller. That's something to really pay attention to. But if we're proceeding as far as we are in cancer research at large, despite this, I think this means we have a huge opportunity in front of us to really maximize our efforts. All right, well, part of that is to say, what does it mean when something doesn't replicate, when it fails to replicate? Well, there's a couple things. One, I think the one that's quick to jump to that I don't believe is the original's wrong. Original's a false positive. I don't think that's the way to jump to. just as quickly, even though we tried hard to guard against it, the replication could be wrong. We could have missed something. We tried our hardest, but we're humans, right? We're doing limited information. The truth is, I think they're both right. I think the answer is that both the original and the replication are correct, and that the difference is we don't understand the conditions. And that's why replication is so important. That's why being reproducible is so important. It's not that you can find the result, it's that someone else independent of you can find the result, and that you know why. You know the conditions that are necessary. This is a brief summary, and there's a paper, and there's, when I share these slides with you, like there's bibliographies, you can look at it. This is kind of looking at a bit of a theoretical, like what does replications mean in terms of the way that we progress evidence? I wrote this with our co-founder, Brian Nozick. On the left, if you think about this, when we're doing experiments, here we're talking about units, treatments, outcome settings. We're really talking about the universe of possibilities to invest a question, right? Why did you choose that model, right? Why are you designing the experiment that way, right? There's lots of variability. There's lots of room to explore in that space, that circle. And you're only exploring one, maybe two or three when you're doing an experiment. But you're hoping when you do a replication that it generalizes beyond it, that it generalizes to, it's not dependent on that specific lot of antibody. It's that antibody. In fact, it's not specific on that vendor. It's specific on an antibody that is specific, right? So these are the types of questions that we're talking about in everything that we do. So on the right, it says, well, if you have a failure to replicate, it just means that maybe that's not true. Maybe it does depend on that lot of antibody. And maybe that's important, or maybe it's not important. But it means that if somebody can't replicate something, you start to shrink the space of how it generalizes, because it tells you there's something probably specific going on. There's hopefully it's not something mundane like standing on your left foot when you pipette, but something really important, which is, oh my gosh, did you not realize that you need this really important factor that was in the media that is from that specific lot? So replication helps expose this. And if you have the same thing, persistent success, which we had, by the way, we had experiments that we could replicate despite all the changes we made, right? It's never the same. And that's what we want. We want to say, I did an experiment, I hand it to you, I tell you what I believe are the necessary conditions, and you see the same result, generally. That is great, because that tells us that we can keep exploring and hopefully generalize that out beyond just one experiment, one lab, one person. So I think the way we do this, and we do this inherently ourselves, but the more that we can do it with each other across, right? Not just in the lab, but across labs, independent replication and sharing these results. We want to know both. We want to know when it succeeds, and we want to know when it doesn't. But more importantly, we want to know the conditions. We want to know all the details I had a hard time finding in this project, because that exposes how much we're going to actually learn. All right, final slides, because I know I have to wrap up. This is a great quote. It's from computational scientists in Stanford back in '95. I think it resonates today. I put in red what I think you can swap out, right? Which is when we write papers, when we do our research, that's a postcard. The real scholarship that we're doing are the reagents we make, the data that we generate, right? Like that's the scholarship. That is what needs to be transparently shared with the community in order for us to progress. And otherwise, no one can recreate that. And that's true when we're thinking about it from a computer science standpoint. It's just as true in cancer biology and other fields. So what can we do? I'm just going to give you a teaser, which I know others are going to get into. One, and Marcus is going to do a better job of me, which is you're part of a social system. We as scientists are just part of a social system, right? We have universities, we have publishers, we have funders, societies. So what can each of those do to help align what we want, which is open, reproducible practices, with what we're incentivized to do, which is publish papers, get grants, right? If those two are in conflict, that's the challenge we have. So the more that we can incentivize these and align those, the better off we are. So anyone that is involved in institutional policy or journal policies or funder policies, this is to me the opportunity we have. And Marcus will talk about that a bit with UKRN, because I think this is exactly what they're trying to do, which is to help align that, make it so that we're rewarding and training and supporting the exact behaviors we want, right? Versus them being accidentally in conflict with each other. At the same note, you as a researcher have something to do, which is try to share more. It's hard, and I'm a big fan of just pick one thing and start with it. Don't let perfection be the barrier. The hardest thing to do is to change your behavior first. So sharing data code, it's the first thing to do. When we write papers, find ways to deposit information in repositories so that someone else can get access to it, whether that be your future self or not. Electronic lab notebooks, I think, are a great way to get it. And especially if you can make those lab notebooks something that you can make openly accessible and then link it in your paper. There's a lot of cool organizations. COS is one of those that are helping push that in terms of electronic tools. I hammered on reagents a lot because to me it was a huge barrier and it's annoying. There's a lot of administrative burden, but like anytime you can deposit something in a repository, there are groups out there that's what they want to do, is make reagents accessible to others. I think that is a great resource we should do more of. So the more that you can do that, especially if you generate one, the easier it is for the next person behind you. And I talked about this concept of pre-commitment, right? The pre-registration registry reports. Registry reports is what we did with eLife. There are a lot of journals and a lot of different fields now that offer this, and a lot of organizations that do this in animal research and not. These are links when I send these slides that you can look into it more. Try this. Doesn't do it for everything. I think this is the question we can have a discussion later on, but especially when you're trying to confirm, especially when you're trying to say, do I think I have the conditions that are necessary? Do I think this experiment is testing my model? Those are times to do pre-commitment. Those are times to really put your cards on the table and say, all right, this is what I think is actually happening. So let me test it. Let me hold myself accountable and lay it all out there and make that hopefully even publicly available so people can see it. Another way to put that is a lot of the discussion about medicine is still around, rightly so, data. So much more beyond that iceberg. There's so much more we can do to improve it. So I will stop there. Hopefully I'm okay on time. Like I said, the slides are available. I'm sure this talk's available and you'll have everything, but a lot of different resources are here. But I really want to thank you for the time and letting me talk about this project.
We're further fine on time and you have time for questions.
Well, to begin with, thank you. That's really an excellent presentation. And it's very clear indeed. Well, what I don't know whether I'm surprised or not, but there are many or too many people that were reluctant to support you, to support your proposal. Don't you think that the results of your reproducibility study, should you have been able to do the job, would have been even worse as compared to what you obtained? Because probably if they refuse to, there may be some reasons.
Yeah, so it's a good question, right? The question, and I've asked this before, right? Which is, we see that there is reluctance of engagement. We can code it, and the question is that influencing the results we have? One, I don't think as much data as we did generate, I don't think we have enough to actually tease that apart. It actually is a bit mixed in what we have. But I do think generally, overall, we probably would have been able to do more. I think we could have changed our model more to accept the fact that not all that information was readily accessible. I think we went in there, even though I knew it a little too naive still, which is, yeah, of course people are going to give us all the information we want. Because of course they have it and they want to, right? The truth is they don't have it and some don't want to. So I think that we probably would have accomplished more if we did have more buy-in. And I think this is this culture element that you're absolutely right on, which is if we can get into this culture of saying, this is normal, right? That we want to share more, we want people to replicate it. Not everything, but especially, I mean, in some cases, this is a great thing. I had people, I think rightly so, say, this is awesome. That's awesome you picked my paper. Like, I know you didn't pick me out specifically, but the fact that my paper is getting attention and that you want to double check it like this is great. And I want to help. And if it doesn't work, that's fine if we don't get the same results. Hopefully we learn it. But I do think you're asking the right question, which is, I do think if we had more people participate, we would have learned more. And that maybe, yeah, it would have potentially been rosier because we would have gotten more information that just wasn't in the paper.
But here there's the issue of biasing the sample.
Oh, it totally is true too. I mean, that absolutely that there is a question of people who don't want to help, that don't want, that there is a chance that probably the results are not there. I mean, do think of really hard. The results I showed you are highly biased. We're not able to do either because we or the authors did something that prevented us from looking at it. So I'm showing you probably the rosiest picture I can, because it's the only picture I can show you, and it's highly, highly biased, which is why I think the challenges are so important. Because if we sat there and I replicated everything 100%, even with that bias, you'd say, oh, don't worry about the challenges. We can deal with that. That's just part of our process. Instead, we're saying, listen, it's 50% at best, 85% reduction in effect sizes, probably worse, right? And nobody's giving you information. So I think you're absolutely right there, but I don't know that. We don't know what our bias is as well, but I do think that's probably true as well. What's that?
Thank you very much for your presentation. It's really exciting to listen to it. I've read the papers, but I was wondering, you were speaking a lot about the culture within a certain scientific community and how it is reflected in the publications, and in particular the fact that there is quite a lot of very important missing information that is absolutely essential if someone wants to reproduce the given study. And there are some domains, in particular computational domains, and this is why we put I guess the quote in the end, where the culture for publications, I'm speaking about the culture, the picture must be not rosy too, but I'm speaking about the culture. The method part is also a result. And so it is viewed as being something extremely valuable in the paper. When you move more toward, the more and more you move towards biology, then less and less method is a result, except for certain papers that are like Nature Methods, publishes methods, etc. But maybe putting more value as a community, it's a cultural shift on the method part could be an incentive for the authors, a positive incentive to provide more details that will help the others. And so I'm speaking as coming from computational side. So what do you think about that? Do you think such incentives can be built?
Yeah.
And what is the carrot?
Yeah, no, it's a great observation. I completely agree with you, right? That some fields, right, computational, and again, there's always issues. We'll ignore those for a second because we're always looking to optimize. But yeah, there are fields and domains, and it's really techniques even. They're very, very good methodology. In fact, that is the point of pride. In fact, chemistry in some areas is great at that, especially when you get to like synthetic chemistry. When we ran into the couple of these papers, my goodness, they'd have pages and pages of methods, really detailed, amazing. Then you get to like the very complex animal experiment, it's like 2 lines. And you're like, come on, like I get that they're both complex, but like that's not true. So you're absolutely right that there's not an incentive. The incentives are shifted very differently. I do think this is where that registered reports model is one to think about, right? That is exactly what that model is meant to do, which is reward on the methodology and the question, not on the outcome, right? Versus right now we reward on what did you find, and it better be positive, right? We want a nice story to talk about. Versus what are you trying to do? What is your methodology? Like, what is the question you're asking? Why you're choosing that? Like, that's what you should reward. So that's one thing I think that can help. And again, there's evidence to suggest that it does work in some fields, like social psychology is one that has some traction. And you can of course mandate it. You don't have to do a carrot. You can do a stick, right? When you think of clinical trials, I'm not sure. I'm a bigger fan of a little bit of both, but carrots are the better way to do it. It's the same problem we have with data. Data has the same problem. It's essentially considered like race. It's not the primary outcome. And I think one thing to do this is to recognize that one, if we have them as citable, so journals are great, method sections are great, making those, but also making that something that you can actually put in your resume, put in your grant applications. That's the movement that also has to happen, which also then should be mirrored when we think about institutions, which is that should be in your tenure promotion packages. So if I'm talking to early career researchers, what I recommend you do is, I even do it myself and I would do it now, which is don't just put in your CV, your papers. In addition to that, also put in other things that you're doing, open data, open methods, right? Highlight, even if it's not perfect, highlight the fact that you're embracing open science behaviors, because I think that's one way to also create that culture, which is you elevate yourself as a desirable candidate as a result of it. But it needs to get aligned with, right, I think journals incentivizing it, but really I think funders and institutions probably have a bigger role than we think. Institutions is just a hard one because it's not like there's one group to talk to. I don't know if that answers your question, but those are the things that I think are the right levers to pull.
Any questions? I have one. You have a mic. It's about the recommendations you made about open science, especially the repositories for reagents, and all of the things that you propose. It actually demands resources, which is a big constraint. I mean, some of it is also like for the methods, essentially rallying from older edition where there was limited space for the paper, and actually now we can use the internet and databases to share that, and it's not a big deal. It's more like inertia in changing the culture. But other things, it requires a lot of resources. And so how can we make it so that those resources, especially when funding is limited, which is I think kind of the problem that open science is dealing with. So how where can the resources come from?
Yeah, no, that's a great question. So it gets to a bigger one, right? Which is our resources are always going to be finite. And actually, I'm very excited to see what happens here because, like, I think years in Europe are way ahead of us in the US as far as I'm concerned. Like, I've done some consulting work with the European Commission, and like the stuff that's coming out of their push towards this is really good. And one question they have is, even on data but methods, all of these repositories is how do you support all of it? And I think it gets to this bigger question, which is one, how much is needed? So there's this open-ended question. And then two, there's questions of like how to do it as efficiently as possible. So, right, when we think about some of these resources, especially when like they're reagents that we already know are important, so you have to have a value proposition that goes up. And then what I want to do is just kind of basically enable those technologies to go at scale. So AdGene is a good example of this at plasmids, which is generally low to like maintain, but they've got a great model that works and they can impact. Animal models, I think, are really, in theory, they are hard, but there are different ways to go about it, right? Maintaining live animals, yeah, good luck on that, right? That's really, really expensive, but we know that we can cryogenically preserve aspects of it. So I think we need to invest more in budgetary lines in supporting these areas of research, right? So that way they're meeting the demand that's necessary while monitoring it. So I'm not answering your question directly because like it is a huge, huge question. I think groups like the NIH in the US are, and the Commission is discussing it. First step is, what's the demand? We know there's a concern. So, okay, so here we know that there's a need. Now the question is asking researchers, well, how much do you produce? How much do you reuse, right? And I think that's the second question. And the third is going to be, well, how do we invest as a large research enterprise like the government to essentially do that?
In your experience, the funders and deciders are already talking about it.
Now starting to. So NIH is behind. I know the European Commission is. They've got some studies, like I said, that I've consulted on where they're not doing anything yet, but they're trying to gather the data. So they've gone out and both data and repositories like this to try to understand across all the different disciplines, because we're talking biology here, but it varies based on discipline, right? Data becomes a huge issue when we think of like geospatial stuff, which is what's the scope of what they need to invest in. So I know that's where the European Commission is, at least in the studies that I've been involved in, which is brilliant because like, at least they're talking about it. I don't know what they're going to do, but they're looking at the scope of the issue to figure out how to invest versus it being more piecemeal, which is the way it's been in the past. And that's kind of the way it is in the US for the most part. NIH has not invested heavily in it. And I wonder if they will. So groups like ours and AdGene, like we actually get support from grants and donors, which is not sustainable either, right? And then let alone if you try to get like a new area, like a new patient center, you know, a patient repository, like who's going to support that? So I think all I'm trying to say is like, I think the more that we can raise this as a concern and a need, and the inefficiencies, which is like, yeah, you know what we're doing is we're going around mailing it to each other, having another industry prop us up inefficiently with all this bureaucratic stuff, and we're still pulling from grants to pay for it. So I think if we also highlight that also can open the door to saying, well, what's a different model? What's a different model to support the research community at large? So I don't have a clean answer for you, but I know that like the best thing we can do is like have these discussions at that right level to try to start pushing for change and pushing for some investment in those areas. It's going to take a long time.
I just wanted to make it's more of a comment than a question. I was really intrigued by your comments about the generalization capacity of the models, and the models that you're speaking is the experimental mouse models or cell lines. And it made me think about there is really a current trend in my research area, which is building AI models for cancer. And the question of the generalization capacity of the models has really become trendy because it's a big issue. If we translate it in your terms, it's exactly the same issue. In fact, it works for this cohort. You change the cohort, you change the city, nothing works anymore. And so the question is, how can the model generalize? And there is no definite answer yet, but at least in AI, there are some possible answers that start to emerge. And one of the main aspect, I think, seems to be the larger the variability in your data. And so I'm guessing in the animals, in your case, or in cell lines, the better the generalization capacity. And so it is very surprising that this, to me, it's striking that there seems to be the same answer for this question, but coming in completely different fields. So
Yeah, that's great. I would love to talk to you more about that. I see it. And if you go just as a quick comment, you can see the same thing. That's why clinical trials are brilliant and why I don't know why. Personally, as a side note, we should do this in preclinical, which is we stage, right? Stage 1, 2, 3, each time increasing variability and increasing sample size precisely to try to get at this question. Also, kind of tempering our resources, which is don't run to like a phase 3 trial until you've gone through phase 1, 2. We should do this in preclinical. Right. And people have made the case, and I agree with it, which is embrace it, but embrace it at the right stage too, which is if you're just doing it in your lab, you're not going to have a lot of variability. If you really want it to have legs, start thinking about multi-center animal studies, for example. Right? Really. So now you just got to engage. It's back to the resource question. I don't do it for everything. So like, how do I know when it's time to go up that ladder and invest more? But I completely agree with you. There's more to get on that. So completely on the right page from my perspective.
Thanks a lot for the presentation. So all the papers and experiments you looked at made it through peer review, right? But I mean, what you did is much more detailed and much more work than what usual people can do in peer review. So can you take away any insights on the peer review system itself? Like looking at some of these papers or experiments, you can say these shouldn't have made it through peer review, or there was not good experiments, or these kinds of insights.
Okay. Oh, that's
peer review is one way to also address the replicability crisis.
Right. Yeah. No, that's definitely another topic. It's a really rich one. If you want, for a quick answer on your question about like what my thoughts are on the process, I think the thing that sticks out to me, and it's hard as a peer reviewer, the way that we've designed it, where everything's focused on just the results and the narrative. Like, we forget the rest. We forget to look at the details. Like, I don't think anybody looks at the method section when they peer review. I'll be honest. Like, at least the papers we looked at, if anybody looked at there, like they should have asked for something. Like, some of these were bare bones. Not all of them, right? Especially when you think about the techniques we're just talking about. Some of those are bare bones. Like I said, like I would run some papers where I'm like, I actually don't understand this experiment. I honestly, I don't know what timeline you use. I don't know what drug dose you use. Like, what are you're presenting me a really exciting result. I don't know what it means, but it's one of many. And I think that's a bit of what's been going on. So I think the big push we have, and when we think about changes, these are papers that tend to be full of dozens of experiments. And at some point, it's just like you overwhelm with, here's just a whole bunch of stuff I found and throwing the kitchen sink in there. And so it's hard to know what to peer review anymore. And I think that's my big takeaway, which is it's just so much that like each of the pieces are kind of, some of those sometimes get lost. And that's essentially what we exposed here. And I don't think the answer, by the way, is, oh, well, it's okay, right? We did it a whole bunch of different ways. I actually think that's still problematic, because if I showed you a finding, 6 different ways, and each time I look at those ways, I have a problem, then you didn't really show me anything. And so I think that's the problem we have with peer review, at least from the little bit I have, which is it's just we're overwhelmed and we're looking at the wrong thing. But it totally makes sense, because that's what we're supposed to look at peer review. What are the results you found? How do I make sense of those? Even though, as we all know as researchers, it's all about what did you ask? What did you design? What are the methods you used? But yes, a whole other day topic.
I have a follow-up question on that, and then we'll for the question. I don't know if you saw Daniel Leskins published this week or last week a proposal that I think it's already in his institution, where the peer review actually occurs before the grant. The peer review for the method. It's kind of like a pre-registration, but it's a peer review of a peer registration before the project is funded.
Yeah.
So it's a proposal that should actually work to solve that problem that you mentioned, that nobody reads at the methods. But actually, after the work is done, it's probably too late to look at the methods anyway. So what do you think about having a board or some kind of control of the methods before the experiments are done?
Yeah.
Projects are conducted.
Yeah, no, that's great. And actually, yeah, very familiar with that. We actually do it ourselves, this type of model. I think it doesn't always work. So I think what we have to start getting into these bigger questions, which is what are the types of research that we're trying to do, and not pretend that it's all the same. There's lots of good exploratory research. We should just be funding exploratory research. I have no problem with that. But when you start to pretend that that's confirmatory research, there's the problem, right? And that's the other thing, real quick back on that note, which is these papers are littered with terms like we tested, we hypothesized, we proved, right? And I'm like, oh, no. And if you did, where's the burden that you really are pushing that harder, right? This is why pre-registration, I think, is a valuable tool when it's the right time, right? Not for every experiment. So back to your question, yeah. So like that pre-registration, the registered reports model, right? We're pushing it even farther into the funder model. So we've done that too. There's different models to try to think about how to do this because you want separation. The models that have been pushed out there is like, how do you get the funder and the journal to be independent, yet work together? So there's a bunch of different ones that are out there. We're actually doing a couple right now with a couple different funders, trying it out as well. I think it's a great idea because it also solves a problem, especially smaller funders have, which is we don't fund a ton of research. It's very specific, and we don't know if it ever gets published. So think about that. When you're a huge funder, you're just like, I'm pushing an enterprise and I'm moving a big ship. When you're a smaller funder, especially when you're really focused on like a specific disease area, you very much want to say, did I make a dent? And one way to know that is, what did you produce and communicate to the research community? Many of them don't know. So I think actually it has value the other direction too, which is it helps give input at the right time, which is really think hard about your methods and we're going to fund it no matter what, and we'll publish it no matter what. It's all about your questioning methods. That has to be solid. But it helps the other stakeholders, which is the reader is very happy. Great. I know that the focus is on methodology, not on outcome entirely. And the funder, especially if they're smaller, is happy because they're like, great, at least now I know no matter what you found, it's going to be out there. And the research community will learn from it. That way they're maximizing their investment as well. So I should really, really like it, but I still think there's a lot to be done to explore the way it works in an operational sense. I think we're early days and there's a bunch of different groups that are trying.
Question here.
You showed a slide about the comparing the success of application between when it's done by the researchers themselves and done by other researchers. And the difference was around 10% most of the time. But the reasons you mentioned about the failure application were mainly because the lack of the data or lack of the communication. So I would expect a bigger difference between them. Why it's not that?
Yeah, well, right. You're talking about the nature survey. Yeah, completely agree with you. Yeah, it's a great thing. And I'm glossing over, but I think you're raising out the right point, which is when I'm saying this, I'm not pointing at a specific researcher in the sense of where they are. I think a lot of the issues we have of lack of data and lack of material sharing is back to my own experience in grad school, which is it's the researcher themselves. They don't know where it is, right? So it's actually the same person. And that's why it's so close, is because our inability to not be able to replicate our own experience actually lead to other people not doing it. And then the reverse is probably true. If somebody else can't replicate it, maybe they can't either because they lack the information. Now, again, it gets to this bigger one, which is it's not everything, right? It's not every single experiment we do. And there's some labs that have amazing practice. And actually, this is another spot, I didn't really talk about it, which is the lab itself is probably the best unit to really instill best practices, which is I know a couple labs that say, before we submit any paper to publication or a preprint, ideally, right, first, they actually have somebody else independently see if they can reproduce everything. It's a computational lab again. I think that some of those computational labs have great things, but they actually don't, it's not that they don't trust themselves, it's that they don't want to make a mistake. And so sometimes I've heard them say, we've actually not submitted because we couldn't do it. So we didn't have access to the data, so we stopped. And so it's the same problem and it's their own experiment. They can't reproduce their own experiment because of the same issues I just had. And that's why they just sit there and keep improving their lab practices to say, well, what can I do to increase our own transparency? Because if I myself or someone in my lab can't replicate it or reproduce it, no one outside of it is going to stand a chance. So that's actually why I think it's relatively close.
Yeah, thanks, Tim. I have a comment about this. Every year is organized something called the 10 years challenge. You are asked to reproduce your own research from 10 years ago. Try this, and you will understand why the difference is so limited. You can't reproduce your own experiments from 10 years ago. I'm pretty sure, and I can bet a lot of money on this. So we have to change the way we produce our own science if we want this to get improved.
Yeah, just as a note, no, I completely agree. There's I like that 10-year challenge, it's great. There's a lab, I gave a talk at Colorado State University in the US last year. And they invited me up because they built a whole their ethics seminar, they're responsible for conduct of research. I think you guys have something like that. They did it for the semester where they basically, largely cancer groups, they would look at the project I did, but they would start with one of the papers and they'd say, okay, well, let's start with the original paper and let's read it and see if we can figure out what was done. And then let's read the replication and see if we can figure out what was done. So it's basically mirroring the story I just told you. And then they did the last thing, which is, now let's pick one of our own papers and see if we can figure out what we've done. And when I talked to both the students and then separately the PIs, they were just like, you couldn't do it. You couldn't do it. And again, it was a hard one to manage because some of the people were there in the conversation, right? And there was one person that told me, it was like, yeah, there was an earlier grad student that's like, I didn't know what you did. I didn't know what reagents you used. And the more senior student who published it said, well, I put it all in the paper. And they're like, but no, you didn't. And then they both looked at it. They're like, oh my gosh, you're right. All those details are gone, right? It's in my head. It never made its way into the paper. So I think you're absolutely right, which is it's a hard, but it's a good reflective exercise to improve your practices.
Well, a small thing, but probably we can spend the whole meeting discussing about your results. Yes, you've selected the I cited papers, so I impact journals. So that's, I don't know if it's right, the cream above the milk. It's something that should be the top level, something on which many further projects are based. Have you been contacted by the editors of these journals? Because they should, because you may reconsider, but it shows that the paper should be reconsidered.
Yeah, yeah, it's a good question. I thought actually you were going to ask me about what about journals that are papers that are not in those journals. I've been asked that before, and it's a very fair question, which is like there are other journals that are published less and focus more on methodology. So like, what do they look? So I think this is just a tease of what can be done. Yeah, in terms of the editors of the journals, we've had contact with them. I can tell you Nature in particular, it's tons of Nature articles. They're always actually in touch with us as an organization, not about these specific papers. We try really hard to be careful of towing that line. The reason why is like, I have no reason not to think that there's poor intention. I'll actually give you an example though of how slippery the slope is though. But instead we have the editors talking to us more about like the same questions that we're having here, which is, well, what can we do? Like, what can we do as the journal system to improve this? And it's amazing to see how that keeps progressing every day. When we were an organization, we had this meeting, we had Nature representatives there, editors, they were like, we're not going to do anything. They're like, no, we don't change. They're like, we still publish our journal in paper. They're like, we're never going to give up, something like that. So how are we ever going to change the rest? Now they invite us to talk to them. They're like, we need to change our policies. We need to think about how to improve things. So it takes time, and I think it takes projects and discussions like this to do it. But I'm actually surprised to see them at a higher level talking about what can we do to improve the process. And again, it varies a bit by domain. Some editors are champions. Marcus Manafu, probably will mention Chris Chambers' name. He's amazing. He really pushes hard, toes the line to cause enough stir to get change to happen. It just takes time, and we need more champions like that. But real quick on that, I said I toe the line about like the papers themselves. There's this one paper, I still don't know what to do, so I'm not going to give you the details of the paper's name, but it shows you the cultural element of what's going on. So it was a paper, they actually shared a lot of stuff with us. They shared us their PRISM files. I could see all their data and their analysis, and it was animal experiments. And but it took me a long time to be able to reproduce the exact graphs they were presenting in the paper. And the reason why is I realized that they were throwing away some data points. They were trimming their data, essentially. And which was interesting because in the file, they actually talked about some animals that died. And I was like, oh, well, that's fine. Your animal died. Put it in the papers. People know that. That's okay. I can deal with that. But I was like, why are you throwing these out? And the reason why it just dawned on me, I was like, oh, I know why. Is because it's published in a prestigious journal, right? And they want really small error bars and a big difference. It doesn't change the conclusion, but it makes the result look bigger. So when I talk about that 85% reduction, it's happening in their own lab. Like, I don't even think and the thing that's important there is that they trimmed their data to get a nicer looking result, even though it was the same result. But they also saw no problem with it because they shared the file freely with me. So I think this means that there's a lot of education that we can do back to like proper use of data handling, proper use of statistics, because it's happening freely within the system and nobody quite knows what to do with it. And it's actually causing some of this, which is we're tricking ourselves by accident because we're trying to make things look nicer than they really are.
You mentioned the impact factor. Do you know what the impact factor is a good predictor for? retractation. The higher the impact factor, the higher the retractation rates. Okay, so this publish or period gain has some side effects. One of this is the retractation. So impact factor should be not a main concern, but when you need a job.
Right. And I think that's the part to change, right? When we think back to the carrots, which is the carrot is not just how many publications you have, and same thing as an early career researcher, right? It's where you publish. I mean, that's all wrong, right? It should really be about the quality of the work you've done. So how do we shift that? How do we shift that culture so that jobs, promotions, grants are all about the quality and the openness of the work and the rigor, not about the name of the journal? And it's hard. It's going to take a long time to do that. And ideally, groups like Nature will actually make themselves be both. So maybe that's what we'll shift to, is the name itself will get back to the integrity that.
I have a question more on it's more conceptual about the notion of replicability. Because you said, in the graph where you compare the effect size of the published results and the replication, and you said in the perfect world, this should be a diagonal, right? On the same, on the other hand, replication is to be, variability is to be expected. Of course, 85% south of the diagonal, it's not to be expected. But so, if we were to define replication and reproducibility in that sense, in a quantitative way, which is something I'm thinking about a little bit lately, how do you deal with it? So we wouldn't expect a diagonal. We wouldn't expect 85% south of the diagonal. What would you expect?
That's a great question. I don't have an answer to. So there's a couple of things, and I've been glossing and using different terms. So one is I would expect high correlation when we're talking about here, like computational reproduction. So like if you publish a paper and you give me some statistics or you present graphs or whatever, I would expect that to be really close. That just tells me that you didn't trim your data, like I just told you, that you have access to data. The question you had before, right, which is in my own lab, I should be able to get the same results from at least the presentation standpoint, which is data, code, paper. So that part I would hope to be high. I also know that it's not, because as you get more and more complex, you get into like machine learning and AI, that starts to become very complex because now the system itself has to be contained. So I say that generally knowing that like, I don't even think that can be perfect because we're going to constantly pushing the boundaries. When we think about what I'm talking about here, which is generation of new data, replicability, right, that term, yeah, I don't know the answer. So, and I don't know if anybody really does know that. That's why it's such a great question, right? This project shows you with all of its own biases that we've talked about, an estimate. And it tells us this is an estimate that we currently have. So now to me, the big question is like, how do you move that estimate and how do you optimize and what should be optimized to? Because what we don't want to do is sacrifice innovation and discovery and being potentially something that can't replicate at cost of just 100% focusing on confirmation. So I think this, to me, in my mind, it's more about not at large, which is what we did here, but more about progression. Early on in a project, I expect us to fail. Later on, as we invest more resources, I expect us to have higher and higher replicability rates. And I expect us to actually start to embrace heterogeneity of the sampling and the strategies to improve that accuracy. So that way we're getting more and more increased accuracy of our estimates later in the process, right? So I'm not quite answering your question because I don't know it in terms of like a quantitative number, but I do think that there's this other dimension of time of progression of an idea that has to be factored. And to me, it's like basic discovery up through translation. Like that's the window we have, which is I'd expect it to improve. Because if not, then we're investing our resources the wrong way, which is also why I'd say make sure that you invest in the pipeline. You want lots of good discovery research, knowing that it's all suspect. Nothing wrong with that. Eyes wide open. And as we move along, increase rigor and be transparent the entire time.
Okay, so I guess this is time for the first coffee break. Of this, we're going to have coffee.